Hello to our friends joining us via recording. Today we are reviewing for the Unit Number 2 exam. As a reminder, this exam opens up first thing Monday morning after spring break, so 12:01 right after midnight, Monday after spring break, and it closes Tuesday night after spring break. So we still have lots of time from this recording until we take that exam, but please make sure to take advantage of your little bit of extra time over spring break. Every now and again, do a little bit of review, but take some time to break as well. For Unit Number 2, we talked about a variety of different things, and to help us review those things most effectively, we did a pretty extensive group work activity today. So we're going to spend a lot of our time now going over what we worked on in groups. The very beginning of Unit Number 2, we started by talking about the levels of protein structure. When we talk about the levels of protein structure, something that we need to definitely be familiar with for exam number 2 is what each of these levels of structure are. So I'm going to ask my friends here in class to help me out in the chat. I want to try to define each of these levels of structure here. So let's start with primary structure. We'll start at the very lowest level of structure. How would you describe what the primary structure of a protein is? What's the easiest description you can come up with for primary structure of a protein? Yeah, so Kira's tossing out the order that the amino acids go in. Yeah, the chain. So their sequence, basically. I'm going to put sequence of amino acids. When I line them up, what order do they go in? That's the primary structure of a protein. Remind me, it is or is not important that my amino acids line up in order. Does primary structure matter? Is it a big deal, small deal? Yeah, it is important, right? It's very important. If we mess up the primary structure of a protein, there's more than a 50% chance that we're going to mess up all of our other levels of structure. Remember that this is what happens in sickle cell anemia. In sickle cell anemia, there is a 1 nucleotide mutation. The DNA changes by 1 letter. And with that 1 letter changing in the DNA, that leads us to make a different primary structure. And if I make the wrong primary structure, I'll probably make the wrong secondary structure and the wrong tertiary structure. And in the case of hemoglobin, which is the protein we're talking about in sickle cell anemia, we have issues even at this level of quaternary structure. So we just defined what primary structure is. That's the order or the sequence that the amino acids go in. Let's jump to the opposite end of the spectrum here. When we're talking about quaternary structure of a protein, is there an easy way to describe that? Or if you were trying to tell somebody what quaternary structure is, how might we talk about that one? Yeah, so Kira is hitting on an important point. Kira is saying when we talk about quaternary structure, it's more than 1 amino acid chain. So in quaternary structure, which is something that hemoglobin has, it takes more than 1 chain of amino acids coming together to build a protein that actually does its job. So hemoglobin actually needs 4 different chains to do its job. Every protein in the body has tertiary structure. Not everybody has quaternary structure. Everybody has tertiary structure. When you're thinking about tertiary structure, remember that this one is my 3D shape, tertiary structure, my 3D shape. I need to make sure that my 3D shape is correct on 1 amino acid chain. That's tertiary structure, 1 chain. Because when I try to put more than 1 together, if one of them's not the right shape, you better believe that the group of them together is also not going to be the right shape. So more than one amino acid chain together, folded into the right shape, that's quaternary structure. One amino acid chain folded into the right shape, that's tertiary structure. Getting into this right 3D shape starts with, or it comes from, to some extent, what's going on with my secondary structure. There were two kinds of secondary structures that proteins can have. What are the name of those secondary structures? Yeah, so you'll see on the exam here, I'll try to draw it for us. You'll actually see the Greek letters for them. I'm going to be all technical on you, and you have that in your notes packets too. Oops. So their names, they are alpha helices, so an alpha helix, and beta sheets. These are the two structures that make up secondary structure in a protein. So an alpha helix, which if you remember, that's kind of what a ribbon on a birthday present looks like. So it curls around and around and around, making that bouncing ribbon. Or a beta sheet, that's a little bit more like an accordion, where it folds on top of itself, makes a flat structure. I need my secondary structures to be correct for me to be able to get my 3D shape, or my tertiary structure, correct. And I need my 3D shape of each chain to be correct for me to be able to put them together correctly. So, big idea with proteins, I need every single layer of structure to be correct in order for this protein to come together. If I mess up primary structure, I will probably mess up all of those layers above it. Hey, by the way, with proteins, true or false, a protein needs to be in the right shape to do its job. True or false, a protein needs to be in the right shape to do its job. Yeah, that is definitely true. It's the whole idea that goes to the name of our course, right? Anatomy and physiology. Yeah, they go hand in hand. The shape of my protein, the anatomy of my protein, is necessary for the physiology of my protein to be correct as well. We have a name for when a protein permanently loses its shape. If I permanently lose it and I can't get back to that shape, now I'm talking about a protein, which here's an underline highlight star word for us. I'm talking about a protein that is denatured. If a protein permanently loses its shape, it's also permanently going to lose its ability to do its job. So if you're denatured, that is the end of the road. It's like those egg whites that we talked about, right? You go from being clear to being white. And like Kaylin mentioned, we can't go back. We can't fix it. So a denatured protein is never going to do its job again. And that's bad news for your cell, because the proteins in our cells all have special jobs. So we don't want our proteins getting denatured. That would be bad news for us. All right, so we've talked about what each of these levels of structure are. We described them. Now we need to think about our parts of the amino acids that are involved in forming these levels of structure. One of my groups asked a good question, and I want to make sure that we say this on the recording here. I'm not really going to hold you responsible for knowing which parts of the amino acids contribute to quaternary structure. So quaternary structure, we didn't talk a lot about exactly how that develops. So when you're studying the parts of the amino acid involved in forming my structures, really what I care the most about, primary, secondary, and tertiary structures. So let's start with primary structure. When we're talking about the amino acid sequence, like we have here, the amino acid sequence, is that something that's dictated by the blue circle part or by the yellow star parts? Who's involved in forming primary structure? Yeah, it's those stars, right? So my stars help me to form primary structure. The stars, as we can see in our picture here, they are showing me the amine group and they're showing me the acid group. Hey, my circle is not labeled. This R group here, what was the word that we used for it? We sometimes called it the R group, but yeah, we mostly called it, the easier word to think about it is the functional group, the functional group. So my blue circle is my functional group, and then I've got my amine groups and my acid groups. Big picture idea here, if the amine group is doing something, the acid group is also doing the same thing. So I connect one amine group to the acid group of another amino acid. That's how I put them in order. That's how I build a line. So the amine group and the acid group, my star parts, help me to put my amino acids in order. Which parts of my amino acid then are used to form those alpha helices and those beta sheets? The curlicue ribbons and the accordions. Do I use the stars or the circles to make my secondary structure? Yeah, the secondary structure is also the function of, or the job of, the amine group and the acid group. When the amine group of one amino acid interacts with the acid group of an amino acid that's a little bit far away, that's how we generate the alpha helices, or that's how we generate the beta sheets. When an amine group and an acid group that are right next door to each other, when they interact, they will form what we call a peptide bond to hold themselves together. When we're interacting an amine group and an acid group that are really far away, they're going to interact with each other using hydrogen bonds. So primary structure, and I'm getting a little ahead of myself, I know. Our next slide will outline these things for sure. But primary structure is where I use peptide bonds to connect an amine group to an acid group. That's primary structure. So when I connect an amine group with an acid group right next door to each other and I use a peptide bond, that's primary structure. When I make a hydrogen bond, and remember we put bonds in quotes, right? Because the hydrogen bond isn't very strong. When I use a hydrogen bond between one amine group and another acid group that's farther away, then I'm building secondary structure. So secondary structure is when amine groups and acid groups that are not neighbors interact with each other. If we're next door neighbors and we interact with each other, that's going to form my primary structure. When we start talking about the tertiary structure, the 3D shape of a protein, is that the job of the stars or the circles? Who gives us that 3D shape? Yeah, the 3D shape, that's the job of those functional groups, the functional groups. Because remember when we talked about functional groups, we sometimes call it the personality type of the amino acid. So remember that some amino acids really can't stand water. They're afraid of water. So those amino acids will group together. They're going to form what we call hydrophobic attractions. They all want to squeeze together. Some of these functional groups are polar, and they'd like to be next to water, or they'd like to form a hydrogen bond with somebody else who's polar. Some of them are going to want to form a covalent bond with each other and attach to each other. So the overall 3D shape of a protein, that's because of the functional group. And remember that the functional group is my personality type of my amino acid. Also a reminder, I don't think I need to remind you this, but I want to make sure we know, please, please do not memorize the personality types of the amino acids. I promise you, I will give you a table on the exam that tells you this one is polar, this one is negative, this one is positive. You've only got so much mental space in your brain, right? And knowing the personality types of amino acids is not something I want you to use that space on. So know that a nonpolar functional group is not going to want to be by water. Know that a polar functional group is going to be attracted to another functional group, or a negative is attracted to a positive. But don't know who's who. We're not going to sort them out. That's for your biochemistry class. That's not for our anatomy class. So 3D shape, functional group. Otherwise, those alpha helices and beta sheets, that's because of the amine groups and acid groups. The primary structure is also from amine groups and acid groups. So we started to talk about this. Remember that there's kind of four main kinds of chemical bonds that are involved in forming proteins. We just mentioned these ones right here. These things called peptide bonds. Which level of protein structure were the peptide bonds involved in? Peptide bonds. Yeah, peptide bonds are the primary structure of a protein. And remember, I told you, anytime you see the word peptide bond, you should be thinking protein. The only kind of molecules that use peptide bonds are proteins. So when I connect together two amino acids using a peptide bond, that forms my primary structure. Yeah, I like that. Kira's mentioning that primary structure and peptide bonds both have P's along with proteins. So the three P's: peptide, primary, protein. I like it. When we talk about forming secondary structure is formed because of hydrogen bonds. So secondary structure, we mentioned this on the last slide. Remember that secondary structure was those alpha helices and the beta sheets. These are going to form using hydrogen bonds. So when one amine group that's far away from an acid group, when they form hydrogen bonds with one another, we're going to curl up into a ribbon or we're going to lay really flat. Secondary structure comes from hydrogen bonds. That brings us to tertiary structure. Remind me in the chat, because I'm forgetful, what is the tertiary structure of a protein? The tertiary structure of a protein. Exactly. Yep, a protein's tertiary structure is its 3D shape. We said on the previous slide that the 3D shape came from the functional groups. So when we're thinking about the kinds of bonds that are used to form that 3D structure, we should be thinking about all the different kinds of bonds that those functional groups could make. So let's say that I am a polar functional group. Polar functional groups, here's everyone's least favorite review here. When we use the word polar, that means that we have dipoles. Polar means we have dipoles, right? We're all dying on the inside because we're talking about chemistry again. So remember that polar covalent bonds, these are not equal sharing. When I say that I've got a polar amino acid, that means that it's not sharing its electrons equally, and that means that we've got dipoles. And dipoles like to form hydrogen bonds. So one of the kinds of bonds that my amino acids will form with each other, those functional groups will form with each other, are hydrogen bonds. So tertiary structure, one of the things that we use to form it is hydrogen bonds between my polar amino acids. Hey, another thing I use to form that tertiary structure is ionic bonds. Ionic bonds form when somebody's positive and somebody's negative and they're attracted to each other. Ionic bonds. So another way that I can form my overall 3D shape is I can form ionic bonds between those functional groups. And Caitlin mentioned in the chat for us another one of those types of bonds that we talked about that we can form between our amino acids, their functional groups. We called them in particular disulfide bonds. Disulfide bonds. That special kind of bonds, its name literally means two sulfurs, disulfide. If we're attaching two of the same thing to each other, they're going to share their electrons equally. Remember that non-polar covalent, covalent always meant sharing, non-polar meant equal. So when I talk about a disulfide bond, we're talking about an equal sharing bond. And that's the other kind of bond. These non-polar covalent bonds are the other kind of bond that I use to make tertiary structure. So my overall 3D shape, which remember is due to my functional groups interacting with each other, they can share electrons equally. They can have dipoles that make us interact with each other. Or if they have opposite charges, they might be attracted that way as well. The three kinds of bonds that we use to make tertiary structure. I want to pause for a moment. Do we have any questions? Or send me a thumbs up. Or an emoji that more directly relates to how you're feeling right now. It doesn't have to be a thumbs up. That's my default. Before I forget, here comes your daily penguin. Let me see if I can find. Here's how we all said at the beginning of class we were feeling. There's our skeleton for the day, right? Hopefully we're not screaming quite as bad after we chatted with our groups. All right, so we've talked about lesson number 5, protein structure. Let's move on to a question from lesson number 6. And I think both of my groups found how Dr. Ollis was purposefully being tricky. Yep. But here's what I want to make sure that we recognize. I want to make sure everyone knows this. You have a question like this at the end of the transcription and translation practice assignment, the ungraded practice assignment. I am promising you 100% that when you take the exam after spring break, you are going to see a question just like this too. So this question helps us to practice. The question on the transcription and translation practice sheet helps us to practice. When you're looking at the exam and the exam asks you which DNA sequences would lead me to add a particular amino acid, notice in this question, I am asking you for DNA sequences. I'm asking you for DNA sequences. Just this statement alone, what are some of the things when our groups caught it? What are some of the things that you were thinking about or talking about when you saw that this statement asked for DNA sequences? What were some of the things that we thought about when we saw this was DNA sequences? Yeah, so we're talking about how we have to do translation, right? DNA sequences, they get translated into RNA. Yeah, the word I was using with one of my groups, we actually, there's this process called reverse transcription, which is probably the easiest way to do this question, to go backwards. So here's something else to consider too. When you look at this question, and this question asks for DNA sequences that lead us to put in this particular amino acid, there are some answer choices that right away we know cannot be correct. Can anyone tell me which answer choices? Let's start with this. Which answer choices do we know cannot be correct based on my question asking for DNA? Who can't be correct? Okay, so Kira's mentioning D can't be correct and F can't be correct. There's one other that can't be correct either. Any ideas what other one can't be correct? Well, the fact that it's a DNA sequence doesn't disqualify A. What about B though? Let's look at B. Yeah, exactly. When I have a question that asks you for a DNA sequence, let's remind ourselves, help me out in the chat, DNA has what 4 nucleotides in it? If I'm building a molecule of DNA, what can I build it with? Yeah, so I can use A, T, G, and C. You know what I can't use when I'm in DNA is I cannot use uracil, right? No uracil, so no U's. A note that I want you to write down for yourself somewhere in big letters and highlight it and star it. Anytime a question asks you for DNA, you better not give me an answer that has a uracil. Right off the bat. And these are tricky on purpose. They're included in here because if we're not paying attention, we're going to say they're right. But I ask you for DNA. So my DNA sequence, I'm asking you to tell me which DNA sequence is going to lead to this amino acid. I know right away anybody with a U can't be an option because we never put uracil in DNA. So look at your question. If your question asks for DNA, do not give me something with a U. Hey, flip side of this, if I had a question that asked me for an RNA sequence, what would I not have if it was an RNA sequence? Yeah, exactly. So if I'm asking you, hypothetically speaking, if I say I need an RNA sequence, you would need to give me something with no thymine, no T. Remember, that is what distinguishes DNA from RNA, is whether we have a uracil or whether we have a thymine. So off the bat, even before I look at my table over here, I can automatically eliminate any answer choices that have U's in them because I need DNA and DNA has no U's. So I can get rid of all 3 of these statements that have U's right off the bat. I know they're incorrect because DNA doesn't have U's. Now I need to figure out which amino acids, which RNA sequences would lead me to phenylalanine. So which ones give me phe on the table? So when we go over to our table and we look at it, here's phe, right? Phenylalanine. Notice that I see the codon UUU and the codon UUC. Now, remind me, these codons that I see right here, are these DNA sequences or RNA sequences? What kind of information is on this table? Yeah, exactly. This table is filled with RNA sequences. And that is true 100% of the time. Even though these letters over here, CAC, We could have that be in DNA. It doesn't matter. This table is made of the RNA sequences. It's all RNA. So here's my two RNA sequences, UUU and UUC. These ones would lead me to put phenylalanine into my protein. So two approaches I can take to this question now. Either I can do reverse transcription, where we start with our RNA and we build it back up to DNA. We go opposite, if you will. Or we can go through each of these answer choices that are left and we can turn them into RNA. These are all DNA sequences because they've got T's or U's, or they don't have those. So I can transcribe them. I can turn them into RNA. So we'll look at it both ways. Let's start by using the sequences. Oh, green's not going to show up. Let's start using these sequences here. We're going to transcribe these sequences. So my sequence here, my DNA sequence, says TAC. If I ask you on the exam, which I am going to ask you on the exam, to do the process of transcription, or to build me complementary RNA, TAC, what RNA sequence is complementary to TAC? Yeah, exactly. AUG. Remember that thymine always pairs with adenine. Adenine always pairs with uracil, because we're going into RNA, and cytosine and guanine always go together. A's with T's, or A's with U's in the case of making RNA, T with A, C and G together. So now let's go to option C. Answer choice C is AAA. What would my codon be for AAA, my opposite? Yep, exactly. It's all about U, right? U. That's the way kids say it. So that's my complementary RNA for answer choice C. And for E, when my DNA sequence is AAG, yeah, exactly. UUC. If we didn't start by looking at the table, if we just started with our answer choices, and these were the three that we did transcription on, now we can go over to our table and we can look for what amino acid these lead to. So AUG, that's my first one here. Remember that with this table, you start in your column on the left side, my first letter. So here's the row that I'm looking for. Everything that starts with A is in this row. Then I'm going to go up to the top for my second letter. My second letter is the column I'm in. So I'm in the first column. So here's my row, here's my column, and my last letter is G. Either I can go over here to the far side, or I can use my literary skills to find where AUG is. The codon AUG leads to the incorporation of something called methionine. Does anyone remember from when we were studying this way back in the day? Why do we care about AUG? This one is actually a little bit special. Yeah, so AUG is called my start codon. Start codon. This is how your ribosomes know that we're supposed to start the process of transcription. So every single protein in your body, the first amino acid that we put on it, on that chain, is methionine, because that's what your ribosomes use to tell them to start the process of translation. Methionine, met, is not the same as phenylalanine, as Phe. We can eliminate this answer because when we looked at it on the table, it did not lead to phenylalanine. My next option, UUU. So we'll go back to our table here. We've got the letter U, first row. Second letter is a U. That's my first column. And UUU. right here leads to phenylalanine. There's one correct answer for me. Now my other one is UUC. We'll go, we had UUU up top. Notice right down below it, UUC, also phenylalanine. So my other correct answer would be this. Now check this out. My question asked for the DNA sequence. The DNA sequence. Notice that the DNA sequence, the two that we picked, they're right next to the RNA sequences you found on the table. I kind of did transcription for you, if you will. I gave you DNA and then I gave you RNA, and DNA and RNA. Please, please make sure when you're taking the exam, read what your question's asking for. Is it asking for DNA or is it asking for RNA? This question would be really easy, right, if I asked you for RNA. If I ask you for DNA, you've got to think a little bit harder, do that one extra step. We mentioned this term reverse transcription, so I'll just toss out there for you the other way that you could do this question instead of going through here and translating or transcribing each of these. What we could have done is we could have said, hey, over here on my table, phenylalanine, there's two options here. So the first option was UUU, and the second option was UUC. Reverse transcription is basically saying, I've got my RNA, let me go back up to DNA to rebuild what the DNA would have said. If we're doing reverse transcription, we're making a complementary molecule to the sequence that we have here. Instead of using U's, we would put a T in there. That doesn't matter for these sequences. So when I'm doing reverse transcription here, I've got a U in my RNA. Well, actually, I've got three U's, right? Which would mean that they would each pair with an A. So my first DNA sequence that leads to that RNA sequence, I can reverse transcribe or build the complementary DNA to it to get AAA, or I can get AAG if I build what's complementary to that. So the other approach that you could have taken, if you wanted to start with the table, you need to build the DNA sequence that gives you these RNA sequences. Either way would work. Either go through and take these DNAs, turn them into RNAs, and then look at the table, or start with the table and go back to the DNA that it would have come from. Kaylin asked the question, if we asked for the RNA sequence, would it also be transcription or translation? So if I ask you for the RNA sequence that leads to an amino acid, I'm basically letting you skip the process of transcription. I'm letting you just go straight to this table and not have to do any extra work with it. So if your question asks you for an RNA sequence, you can use the table as is. We've already done the process of transcription. We don't care about transcription. We're just caring about translation. I know this one was tricky. It was tricky on purpose. I wanted to make sure we did a tricky question like this together so that it doesn't surprise us when it shows up on the exam. How do we feel about this question now that we've talked through it? Okay, so I'm getting some thumbs up, smiley face. Again, you can use the transcription and translation practice page that's located in the Lesson 6 folder. There's another question like this. And I know that one of my groups too was going through and rewriting this question, if you will. You could totally do this on your own when you're studying as well. Maybe instead of phenylalanine, maybe we want to pick a different amino acid and we want to predict the answers for that. So you can go through and change up this question, use that practice sheet, use this practice question. Just read your questions slowly. I can't emphasize that enough. When we take our exam, read the questions slowly. Look for any clues you can find to help you as you're working on it. All right, let's shift gears. We are shifting gears now from talking about lessons 5 and 6 to jumping into lesson number 8. And I know for several of us, lesson number 8 has not been our top priority because we had the lab exam and we had lesson number 7 to wrap up. But let's do a little bit of a review about lesson number 8. Lesson number 8 was about the skeletal system. And the way that we form most of the bones in the skeletal system is using something called endochondral ossification. Endochondral ossification. Let's remind ourselves when we're talking about this process here, remember that the word ossification means bone building. That's ossification. Remember that the endo part of its name was inside. What's that chondral part of its name mean? Chondral. Yeah, exactly. Chondral means cartilage. Remember, chondrocytes were those cartilage cells. We talked about in lesson number 8, chondroitin sulfate. That's the squishy stuff outside of cartilage. Chondro always means cartilage. So endochondral ossification. This is the process that we use to build most of the bones in your body. We do not build which kind of bones. There was one particular group of bones that we don't use this process for. Who do I not use this for? Yeah, exactly. I don't do this for flat bones. So not for flat bones. Not for flat bones. Everybody else, we build with endochondral ossification, especially your long bones. That's the big one that we build this way. Endochondral ossification for those long bones, not for flat bones. We talked about that's bones like the sternum, that's bones like the ribs. I was chatting with one of my groups. That's also a lot of those skull bones. We don't use this process for those flat bones. When we talk about this process, inside cartilage bone building, when does this process start? Endochondral ossification. When does this process start? Yeah, so we've definitely got an issue here with this word after. I hope your bones are forming before you're born. Otherwise, you have nothing for your muscles to pull on. So absolutely. The process of endochondral ossification, it starts before a baby is born. Yeah, so several of us are mentioning in the chat, it starts around 9 weeks. 9 weeks gestation is when we first start this process of building bone inside a cartilage model. When we do endochondral ossification, we don't turn all of the cartilage into bone right away. We actually don't ever turn all of the cartilage into bone. I was chatting with at least one of my groups about this. What kinds of cartilage do we not turn into bone right away? Where do we keep cartilage around? Yeah, so it is hyaline cartilage. That is correct. Yeah, one of the places where I keep my cartilage around, and I keep it around actually forever, is a place like Anisha said for us, articular cartilage. So that's there forever. We never turn articular cartilage into bone. This is the kind of cartilage that's at the joints. So there's always going to be cartilage at the joints to help those joints move smoothly so that there's not rubbing. Articular cartilage never turns into bone tissue. The other place, like Carissa mentioned for us, the other place that I don't automatically turn into skeleton, or into skeleton, into bone tissue in the skeleton is the epiphyseal plate. The epiphyseal plate. We keep that around until about when? How old are you when that epiphyseal plate goes away? Yeah, so somewhere in the ballpark of 18 to 21, typically. 18 to 21 years old, it goes away. So the epiphyseal plate is another place where cartilage sticks around until you're about 18 to 21. At that point, we do turn it into bone tissue. What do I call it when it's turned into bone tissue? If it's not an epiphyseal plate anymore, what did I turn it into? Yeah, exactly. If it's not the epiphyseal plate, it's the epiphyseal line. The epiphyseal line. So I don't turn all of the cartilage into bone, not right away, or in the case of the articular cartilage, not ever. It always is going to stick around with some cartilage left. We've mentioned this in the chat. Want to make sure I clarify it though. When we talk about the fetal skeleton and we talk about turning cartilage into bone tissue, what's the kind of cartilage we're talking about again? Endochondral ossification. Which kind of cartilage are we turning into bone tissue? What's the fetal skeleton made out of? Yeah, exactly. The fetal skeleton is made out of hyaline cartilage. Hyaline cartilage. So hyaline cartilage makes the fetal skeleton. It also makes up the epiphyseal plate. It also makes up the ends of bones, that articular cartilage at the ends of our bones. All right, let's go down here to our next statement. And this statement we can kind of see relating to the statement below it because both of these statements are talking about ossification centers. Ossification centers. So we've got a secondary ossification center and a primary ossification center. Let's start here with the primary ossification center. Where do we find primary ossification centers when we're talking about the parts of the bone? Yeah, so it's a mean word for me to make you type, right? When we're talking about the primary ossification center, we find that in the part of the bone called the diaphysis. The diaphysis, which in easy person words is the shaft of the bone, the long part of the bone. The primary ossification center is found in the shaft of the bone. The secondary ossification center, and there's actually two of those, right? The secondary ossification centers are found in the epiphyses, found in the ends of the bones. So first way that we would consider fixing this question, we are not going to find secondary ossification centers in the diaphysis. That's not correct. We would find secondary ossification centers in the epiphyses. But actually, there is a different way that we could correct this statement. Let's assume that we are talking about chondrocytes and they do live in the diaphysis. Something is making them die. What did we say that I formed that when I form it, it makes them die because it cuts them off from their nutrients? Put a little box here. The other way I could fix this, yeah, exactly. The other way I could fix this is those chondrocytes die when I form that thing called the bone collar. The bone collar. Remember when we're talking about endochondral ossification, first we build a cartilage model that has those cartilage cells inside of it. Then that model gets big enough that the ones on the outside build this bone collar. And when we put the bone tissue on the outside, these cells start to die because bone doesn't let nutrients through. So the chondrocytes die when I put a bone collar on the outside. They're sending out their SOS, if you consider it that way. They're sending out a signal, we need help. In come the blood vessels. The blood vessels form what's called the primary ossification center. So they bring in oxygen and nutrients and osteoblasts, and they start building bone in the shaft of the bone. This process continues, starts around week 9, and it's going to continue all the way up until a baby's born and keep going. But after a baby's born, or right around when a baby's born, we also form those secondary ossification centers. The secondary ossification centers form in the epiphyses. They form in the ends when blood vessels come in there. I don't ever put a bone collar on the epiphyses of the bone. I only put a bone collar on the diaphysis. I only build it on the shaft of the bone. So the diaphysis, the shaft of the bone, that's where those primary ossification centers are found. That's where the bone collar is found. The epiphyses, the ends of the bone, that's where those secondary ossification centers are found. Thumbs up or questions about these statements? Timmy Teo's liking the bone collar. Good. Caitlin, happy to see that I'm blowing your mind. That's my job. Yeah, so asking about the order. Let me see if I have that slideshow. Let's see. Let me check one other place. All right, I will share my screen with you and we can look at this together. I unfortunately, well, maybe I will be able to write. We'll see. Okay, so this should be what you see in your notes. Can we confirm? Is this what it looks like for you all? Perfect. Okay, so we've got a list of steps in here. So we'll go through and let's work on the order that they go in. We're doing endochondral ossification, inside cartilage bone building. For us to be able to do that, we have to start with cartilage. If I don't have a cartilage model, I can't build bone on it. So let's see if it's going to let me. Let's see. Oh, nice. Okay. So step 1, first thing I've got to do. Okay, that's fine. Thank you. First thing I got to do is I have to build my model. Step 1, build my hyaline cartilage model. When that hyaline cartilage model gets bigger, we start to develop that thing that we just talked about before. We start to develop our bone collar. So we build the model and then we put a bone collar on the outside of that model. We put that bone collar out there and we've cut off our nutrients to the chondrocytes. So the chondrocytes that are inside that cartilage model, they start to calcify or they start to die because they're being surrounded by calcium. They send out their SOS message. That causes some blood vessels to come into the diaphysis. When the blood vessels get into the diaphysis, into the shaft of the bone, they bring with them the type of cells that can build bone tissue. They bring with them the osteoblasts. Those osteoblasts make us a primary ossification center. We build bone, going through the process until about the time of birth. At that point, those blood vessels go into the epiphyses, into the ends of the bone. They bring with them more osteoblasts. Those osteoblasts form the secondary ossification centers. And once I've built those centers, I'm going to ossify everything, all of that cartilage, except for the articular cartilage. So my last one, number 8. How do we feel about this? Okay, so there's some issues with the guided lesson. We'll make a note and I'll look into that to try to get that clarified. Put a note, guided lesson 8. Yeah, so this is the correct order. I will check on that guided lesson and see if I can't get it fixed to this for you. So I apologize about that confusion. Yeah. All right, so the one last thing I want to do really fast, since we did it in group work, is we have one more slide with information on it that relates to those disorders that we are learning about. We may or may not have started learning about them yet. We need to learn about them by the end of spring break. So let's do a quick overview to the 3 main bone disorders we're learning about this semester. Those bone disorders are osteogenesis imperfecta, osteoporosis, and rickets. Let's start with this one because its name is really helpful for us. Osteogenesis imperfecta. Osteogenesis means building bones. Imperfecta means not perfectly. Osteogenesis imperfecta. This is an issue with the ability of our body to form collagen correctly. So osteogenesis imperfecta is an issue with collagen. When you have, this is a genetic disorder, when you have a mutation in your gene for collagen, it can be very severe to the point where you can't make any collagen that's very strong at all. Collagen is really what makes your bones not break. So in osteogenesis imperfecta, the easy person name for it is brittle bone disease. Your bones break very easily. Because osteogenesis imperfecta is an issue with the way that your body builds collagen, a protein that we see everywhere in the body, throughout the body, osteogenesis imperfecta is not curable. If we were to cure it, we would have to go in and we'd actually have to change your DNA, because this is a problem that starts down at the DNA level. I don't have the right directions to build collagen correctly. So osteogenesis imperfecta, brittle bone disease, this is something that we can't cure. It's an issue with how we build collagen. Hey, osteogenesis imperfecta, do we typically see this in children or adults? When is this typically diagnosed? Yeah, this is absolutely going to be diagnosed in children. You will be watching a video about a child who was born with osteogenesis imperfecta. It's a video, it's called Our Baby Made of Glass. Yeah, it is really, really sad. But also her parents are very hopeful, which I love, seeing how hopeful her parents are. So getting an idea of what it would be like if you had a child who had osteogenesis imperfecta, where the collagen is formed incorrectly. You watch another video about the other condition that we see in children. In particular, this condition was talked about in Canada. The video actually comes from Canada. What was the other condition that we said was typically seen in children? Yeah, so the other one that's typically seen in children is rickets. So rickets is a condition, it's not an issue with collagen. Rickets is actually an issue with calcium. So rickets is something that kids can develop when they're trying to build bone tissue and they don't have enough calcium. There's a couple of different reasons they could not have enough calcium. First reason is that they just don't have enough calcium in their diet. That's not typically what causes rickets, though. A lot of times, especially in Canada, the issue with rickets is someone doesn't have enough vitamin D. So vitamin D helps your body to absorb calcium. If you don't have enough vitamin D, you can't absorb the calcium in your diet. So treating rickets actually typically comes down to just increasing someone's vitamin D, giving them a vitamin D supplement. Or this is why when you're at the grocery store, sometimes they call whole milk vitamin D milk, because it has extra vitamin D to make sure that kids can absorb that calcium. So rickets is an example of a bone disorder that we would say is curable. It's something that we have ways to treat it so that it doesn't affect you for the rest of your life. Our other disorder, osteoporosis, I know there was some debate amongst my groups if we would say that osteoporosis is or is not curable. I think most of us settled, I would say osteoporosis is not curable. It's something that you can try to prevent from developing, but once it develops, you kind of have an issue. So osteoporosis, the name of it means bones that have holes. That's literally what osteoporosis means. Osteoporosis is also an issue with calcium. So osteoporosis is also a calcium issue. In the case of osteoporosis, your body steals calcium out of the bone tissue to keep the calcium level in your bloodstream high enough. So we're breaking down the bones, leading us to not have as much calcium stored. That will also make your bones more likely to break. Not for the same reason as osteogenesis imperfecta, but they would also break as well. Osteoporosis, pretty much across the board, only seen in adults. I think you read a little bit about that it is possible to see it in children, but pretty much when we talk about osteoporosis, we're just talking about it in adults. Yeah, so I saw some comments in the chat about whether or not calcium would be helpful for various things. Totally correct that administering calcium would be great for rickets as long as it's paired with vitamin D. If you don't have enough vitamin D, you can't absorb that calcium. So calcium and vitamin D, awesome for both of these things, both for rickets and for osteoporosis. Yeah, Timmy Teo, you have a question?
Yes, I have a question. So my question is, does spina bifida fit in all of this or no?
So spina bifida, we will actually talk about in unit number 4. That one doesn't have to do with the bone itself as much as the spinal cord. So when we talk about spina bifida, the original issue that we have is the connective tissue that protects the spinal cord doesn't fully form correctly. So sometimes we are missing some bone tissue down there, but it's not an issue with calcium or collagen. That's an issue with a different part of the prenatal supplement. I believe it's folate or folic acid. If you don't have enough of that, you don't form the vertebral column correct. So it's not a calcium or collagen problem, but it is, we're missing some bone tissue. Yeah, good connection to make. So Fanshan, I see your note that you're having issues with the treatment. So the one thing I'll mention as you're trying to figure out what the treatment for the various disorders would be, the parents of the daughter with osteogenesis imperfecta, they literally say in the video, people come up to us all the time and they say, like, have you given her some milk? Calcium is not helpful for osteogenesis imperfecta. So when you're thinking about would giving calcium supplements work for things, it would work, it would help with rickets, it would help with osteoporosis. It's not going to help with osteogenesis imperfecta. Yeah, like Kira mentioned, they were so over those comments. Yeah, that was the context of their comment. They're like, oh yeah, people come up to us on the street and they're like, Do you ever give her milk? Oh, thanks. We haven't thought of that. That's one of the best parts of of that video. Absolutely. That and when they take her to the park. I'm just saying, like, my mom heart was so happy seeing the little girl having fun at the park. So, all right. Well, I have kept you over time today. I apologize. I hope that our review session today has been helpful for you, that you feel like you've learned some stuff, that you're maybe on slightly better footing. Remember that we have the review assignments. So the first assignment is part 1, that covers lessons 5 and 6. The second assignment covers lessons 7 and 8. And I think it also has some stuff about those skeletal system disorders in it as well. If someone could do me a favor, I think I have people here from both of my lab classes. Please send out a message in your GroupMe to remind everyone that there are visible body assignments and the lesson number 7 assignment that are due tonight. I'm going to try to post an announcement to remind everyone, but if someone could shoot out into your class GroupMes just to remind everyone, lesson number 7 due tonight for lecture, the skin, and then week number 8 assignments for visible body also due tonight. Everything else due after spring break, so Sunday after spring break, but we got to get those things wrapped up before we peace out, at least for a day or two. Yeah, Kaylin's mentioning that she has a lot to work on this week. I suspect, Kaylin, that you are in good company. Yeah, so to clarify, Carissa, there is a visible body assignment for lab. There's 2, I think. And then there is also a the lecture lesson number 7 assignment. So the Blackboard assignment for lab is due after spring break. The visible body assignment is due before spring break. Okay, so if everyone is in the same GroupMe class, I will just make sure to send out to my other one to try to get them, make sure that they know that they have those assignments due today as well. All right, well, I hope you all have a wonderful spring break. I will miss chatting with you, but I will also enjoy having a bit of a break, right, from staring at the computer for so long. So I hope you all can take at least one evening or longer if you can to relax a little bit, but please feel free to reach out via email. Otherwise, we will plan to come back together as a class on Monday after spring break at 1 o'clock. We'll review for the exam together one more time, and then hopefully we will we'll rock that exam. So good luck with your studying. Have a great spring break. I will see you all in a week and a half, forever from now.